Thanks a lot for the invitation, first of all, and for the nice introduction. I will talk today about, three parts. First of all, I will introduce my group and what we were doing so far. In the second part, I will give you a short introduction about plant pathology and what's going on in this co-evolution between the pathogen and the plant, because I think this is a necessary theoretical basis, which I would like to share with you before we get to how I want to integrate into PhenoRob and what I think I can contribute to. So I hope you all hear me. So the title was Learning from Biotrophic Pathogens, but basically these are three parts. So in my group, we are working on plant biotrophic pathogens, and mainly we are focused on fungal pathogens, which can be genetically manipulated, and among them, the major tool we are using is basically Ustilago maydis. And Ustilago maydis causes these dramatic galls on all aerial parts of the host plant. And if you zoom into this interaction, you see here in red the fungus, and in green you see the plasma membrane of the host plant. And I hope you can appreciate that it's a very intimate relation here going on between the plant and the pathogen. And in order to control this interaction from the pathogen side, it needs to secrete small molecules, so-called effectors, which are necessary to suppress plant defense responses, but also necessary to basically reshape the host metabolism and to reshape the development of the plant. As you can see, there are galls formed, and this is not natural behavior of the plant, but it's basically a manipulated behavior due to the action of the fungus. In my group, we are focusing on these effector molecules with the goal to basically, based on these effector proteins, we want to understand their function, and via the functions, we want to get an insight on biotrophy, what is going on during this interaction, really. And the final hope is to use these effectors as molecular tools to manipulate plants as well. So we want to apply our effector knowledge. There is the good news and the bad news about effectors. First of all, the good news is that they are unknown proteins, they are hypothetical proteins, so there's a lot to be discovered. And the bad news is that they are unknown proteins and hypothetical proteins, and because of this, it's very difficult to study them, because from the sequence, you cannot really deduce any function, but you need really to go into experimental approaches, into the wet lab to really test what they are doing. And therefore, we developed a pipeline, which we call effectomics approach, where we do systematic localization, interaction screens, as well as functional screens. And by overlaying these results, we try to form a working hypothesis what an effector could be doing. And we then study specific effectors and develop them. This is now ongoing already since many years, 7, 8 years in my group. And so the results are now popping up slowly. We have identified, just now published in this month, in New Phytologist, one effector which is suppressing basically a co-repressor in a conserved co-repressor, and thereby release an antagonistic pathway, a transcriptional pathway in the plant. We have one story at the moment under revision where we have an effector which is suppressing ROS-burst, one early defense response of the plant via transcriptional manipulation of the host plant. We have one paper in review, which is on a whole cluster of effectors which are suppressing also one of these early defense responses of the plant, the pump-triggered immunity response. And so we are now basically harvesting the fruits from this kind of approach. But the idea was not only to study effector functions, but also to use effectors as a tool. And this is just to give you a glimpse here. We have identified by different screens effectors which can indeed manipulate the plant morphology by just expressing them in the plant. And so these can become very useful tools in order to manipulate the plant morphology or development as well. On the other hand, another line of research in my group is to develop new technologies. And this is a work which was done by a PhD student of mine still in Vienna. And the challenge was here that if we want to study hundreds of fungal mutants, effector mutants, to understand which effector has which impact on virulence of the pathogen, we have to infect plants. And what is usually done is that we then score symptoms. And what you see here is a symptom scoring scheme where we basically classify infected maize plants according to their symptom severity. And this is then done with hundreds of plants after infection with a mutant strain of the fungus versus a wild type strain of the fungus. And then we try to assess, is there a difference between the mutant infected and the wild type infected maize plants? And thereby we deduce basically, is this mutant or this gene which is mutated, is it relevant for the interaction with the plant or not? So it's a very, let's say, laborious approach, and it's qualitative and subjective, this kind of phenotyping, and it's indirect because we just look at the symptom development, but we are not looking at the fitness of the fungus itself. So we were not happy about this kind of approach at all, which is the classical approach in my field. And so we decided to go for a new path. We wanted to develop something which is more quantitative, which is time and space efficient, and which will allow us basically to screen hundreds of mutants at the same time. So we call it pooled virulence assay. And just to briefly give you an idea, so this is a published method now. We basically, in a proof of concept study, mixed 200 mutants which were generated by homologous recombination within the genome, and we infected them as a pool inside the plant. And then we isolated the genomic DNA, which contains, of course, maize DNA mainly, and then fungal DNA. And this fungal DNA, each mutant is basically identical to the other mutant, except for this single point where the insertion cassette is integrated in. So we basically have mainly the same sequence information except for this insertion site. Now, you don't want to sequence millions of genomes. You want to find out how many of which insertion site can be found in this pool. And this was then done by a strategy which we developed with a TN5 tegmentase. We could fragment the DNA, and with a specific PCR approach with bialternated primers, we were then amplifying specifically only the flanks. And by this, we can basically count in the end the reads, which are equal to unique flanks. And this indicates then which mutant was proliferating within the plant and which mutant was disappearing inside the plant and was not fit enough to infect. So it's basically a negative selection screen where you have an input, a selection, and an output. You can compare it with a sieve. You put in all kinds of different seeds, which are having different sizes. And then you have basically, after the selection, only the small seeds left. And before you know what is put in, and then you know how many kilograms of small seeds were inside this total mixture. Or if you translate it into our assay, we start with a mutant library of effector mutants of Ustilago maydis infecting the maize plant as a selection criteria, if you want. And then we look at what is left after the infection, which mutants were successful and could proliferate and which ones were extinct during this selection process. So by this methodology, we could identify not only, so what you see here is basically the 6 independent experiments, and the color code indicates the fold change within the population. The more red you see, the less of it was found after infection. And if you zoom into this area here, these are basically all effector mutant candidates which disappeared after infection, and there are known ones, so controls within this selection, and we identified 24 others which new effector mutants which were not known so far to be relevant for virulence. So why is it important to understand which factors are virulent, which are important for virulence? This gives you a new set of targets on the pathogen side, which you can target in order to restrict the pathogen's infections. So it's a useful way of identifying new targets, molecular targets, to block a pathogen. So this is now work which is going on. So what we then went on is to develop a way to generate in Ustilago maydis not only few mutants by homologous recombination, but thousands of mutants with the help of a jumping transposon. So transposons are basically elements within the genome which can jump from one side to the other, and they do this after a certain, you can activate this activity basically. And so we developed this strategy in Ustilago, and what you see here is a sequencing result of a library which we initially generated, where we could show that there are 7,000 mutants generated in one shot just by this strategy. And each peak, these are reads mapped on the chromosome 1 of Ustilago maydis, is indicating one insertion mutant. So with this methodology, we now can generate thousands of mutants in Ustilago maydis, and the next step will be now to infect maize plants again with a whole genome mutant library and identify basically all factors within the genome which are relevant for virulence. Now I told you I will switch to the second part of my talk, where I would like to give you a short introduction into plant pathology and the plant immune system and the co-evolution with the pathogen. So if a plant is basically recognizing, or if a microbe is coming close to a plant, what happens is that the plant usually recognizes this microbe via highly conserved molecular patterns, so-called PAMs, Pathogen Associated Molecular Patterns. This could be, for example, the flagellin within the flagella of a bacterium. It can be EFTU, elongation factor, which gets released and which is highly conserved. So that these are highly conserved molecules which are common to many microbes, or a cell wall component like chitin for fungi. And these receptors are perceiving these signals, and they are triggering specific defense responses, early defense responses. And these early defense responses need to be overcome by any pathogen which wants to be successful on the plant. And this is done by the help of these effector molecules, as Ustilago maydis uses them, but also bacteria use them, and also pathogenic or parasitic plants are using them, nematodes are using them. So all kind of pathogens are using effectors, but be careful, effector is a term which is just a term for a molecule which is secreted and translocated or acting on the plant side, but it doesn't mean that they are all identical. They might target the same targets, but they're all individual molecules. So this is what is happening in for a successful pathogen. It needs to suppress these early defense responses. So the plants have evolved over the millions of years, of course, ways to still fight back. And this is done by the help of a second level in the immune system of the plant, which is called, or these are receptors within the cytosol, which are recognizing effectors. So these are these NLRs, or for breeders, these are R genes. So these nucleotide binding domain leucine-rich repeat proteins are either recognizing specifically an effector, so they bind to the effector, and this is then activating a defense response, a very strong defense response. Or there are also so-called guard NLRs, which are basically monitoring the homeostasis of the metabolism, and if this gets changed by the action of effectors, they get alerted, and this is then also leading to a very strong defense response. So these are the two layers of the plant immune system, if one wants to oversimplify it a bit. And this was put together in a very nice model, which is basically integrating, on one hand, this co-evolutionary concept over time. And so you can see this is called ZigZag model. It's very commonly known among plant pathologists. You have on the y-axis the amplitude of the defense reaction. On the x-axis, you have basically evolutionary time, if you want. And we start out with highly conserved molecules from the microbes, which get recognized by the plant, and this leads to pump-triggered immunity, PTI. But then effectors evolve by the pathogen, which are suppressing these early responses. Then we get a stronger response recognizing effectors or their action. This leads to effector-triggered immunity. And again, there are additional effectors evolving, which are suppressing these early responses. And this is ongoing forever, basically, this kind of co-evolutionary warfare. So this needs to be kept in mind, that we are in an evolutionary process also on the field in agriculture, and that basically there is never a long-term solution in a way that forever we solve a problem, except if you extinct a microbe. But basically, we always think about how to overcome a certain problem, but evolution will fight back at some point. So now getting more towards the phenorob project, plants cannot run away. And so they are stressed either by abiotic stresses or biotic stresses, and they need to handle them in their way. And they are handling them quite successful, as land plants are covering most areas on this planet, maybe except for the Antarctic. You can find anywhere, maybe even there, you can find land plants. So which factors influence basically the plant development and productivity? This is of course important if one wants to understand what harms our crops on the field. We have environmental stresses, and it depends on the severity of these stresses, on the duration of the stress, on the number of exposures, as well as of the combination of stresses, how severe these stresses are for a plant. And it also depends on the second level on the plant characteristics. This is the second, this green round thing here. So we have the genotype, first of all, the genetic capital, basically, which is there for the plant. And then on the other side, we have the organ or the tissue which is getting infected. This matters, of course, and also the stage of development. If it's at a very early stage, of course, it might kill the plant. If it's just infecting an old leaf in the end of, in a senescent state, it might not be problematic to be infected anymore in terms of for the productivity of the plant. And these factors are then basically integrated, and then it depends basically if we get a response which leads to resistance or which are not sufficient, and therefore it's leading to susceptibility towards this specific stress. And this leads then either to a loss or survival and growth. So what I could grab from the phenorob concept is basically that if we put it together as a general aim towards a goal of a sustainable agriculture, we have basically ways to detect specific stress events in the field. And these detectable events need to be then interpreted in the correct way. And then in the end, the goal would be to find a specific reaction to the specific spot within a field. So you have an individualization basically on the field and a much closer eye on your crops in that way. So the challenges are, on one hand, to detect, first of all, the events, and bigger challenges even to, in my eyes, to interpret them correctly. And the next step, which is also not trivial, is of course to react in an appropriate way in a sustainable manner. So this all together is the goal and the challenge, and how to put myself into this thing. So relevant for the goal number 1 and 2 means recognition of the event and correct interpretation. The challenge is also to identify it as early as possible, I guess. And the question stays in the field, of course, how specific are these symptoms which we can observe by different means. So plants themselves, they sense broadly their environment. This is known, and this is how they react to their environment. This is what makes them survive on the field. So they can sense all kinds of nutritional limitations. They can react to salt stress, they can react to drought, they find out if they are under anaerobic conditions, under extreme temperatures. Biotic stresses can be differentiated into biotrophic or necrotrophic pathogens, because the plants respond completely different to the different type of pathogens. And this can be reflected in transcriptional profiles which have been taken for many plants actually in the meanwhile on different stresses. Here just one example. The blue color indicates basically a switch on of a specific regulatory element, a promoter if you want, which is indicating that under a specific stress this promoter gets switched on and the plant recognizes it, or you can use this as a reporter. And on the other hand, there are publications out there where reporters have been proposed, which are in vivo reporters. So this beautiful beta-lain reporter has been recently published, where basically if you switch it on, you get basically red plants. Very easy to detect, of course. And the one possibility here, back to our PhenoRob goal and challenge number 1, how to recognize an event, could be to spread into the field. And first, of course, this has to be developed in principle, lines which are recognizing specific events and which give you an easy readout, so that challenge number 1 and challenge number 2 can be easily recognized. So you have these red spots would indicate a reporter line which is spread inside the field. And this would allow also a much more easy interpretation of the data set. In the long term, or the last part, of course, the ideal solution is if the plant itself reacts and that no interference of humans is needed anymore. And in general, plants are reacting on a signal anyhow. So if we look at biotic stresses, biotrophic and necrotrophic pathogens, for example, the plants will react differentially. And one idea could be that we screen molecules which have not been used by plants so far in their evolutionary time, at least not these specific molecules, but which are used by other organisms in order to fight against microbes and pathogens. And so an idea would be to screen basically the hundreds of possible antimicrobial peptides which are published and out there, found in various species, and to bring them into the plant to test if we can use them as a response pathway after recognition by the plant in order to basically fight back the specific pathogen. So we will have basically a reaction cycle where we get a recognition by the plant, and in the end also a solution of the plant itself. And so this would cover my contribution to challenge 3, would be to take out human interference at all, ideally. I'm aware that it's not easy and also challenging, but this would be basically the way how I see myself, where I can contribute. And with this, I'm at the end of my presentation. I'm now in Nusaleh 9 to be found. My research has been funded by various sources in the past. And I would like to thank my current lab, my growing lab at the moment, under quarantine situation with masks, but I hope this will be soon over as well. And I'm open to your questions.